What we present here is a robust and simple method to analyze microtubule dynamics using live cell spinning disk confocal imaging in cells synchronized in prometaphase, and the images are afterwards analyzed on a MATLAB-based platform.
The use of red-shifted fluorescent protein in combination with spinning disk microscopy reduces phototoxicity. Therefore, a larger number of cells can be imaged within the same preparation.
Microtubule dynamicity is altered in a large number of pathological conditions. Therefore, understanding the regulation and the behavior of microtubule dynamics in those processes can help us understanding the mechanisms of disease and eventually developing therapies to compensate for them. And the method is also upscalable, so it could potentially be applied in a drug discovery effort.
The method can be modified by changing, for instance, the synchronization protocol and obtaining cells from different phases of the cell cycle. This can be useful when screening drugs against microtubules and identifying the effect on dividing and non-dividing cells. It is necessary to optimize the transfection protocol and seeding density for each cell type. The expression level of EB3 should be low in order to detect single-growing microtubules.
Start by rinsing asynchronously growing HeLa cells in DPBS. Then incubate them with trypsin EDTA for 5 minutes at 37 degrees Celsius. Stop the trypsination by adding RPMI 1640 medium supplemented with 10% heat-inactivated FCS at a 3:1 ratio of the added trypsin EDTA. Calculate the concentration of the cells according to manuscript.